Right, so this is the homologous pair of chromosomes. This contains the maternal and paternal chromatids that are joined together at the centromere. So the centromere is in the middle, how they're joined, and then as you can see there's a paternal and a maternal chromatid. So where the black lines are is where these are both alleles. They both code for the same characteristics and they're found at the same locus on the chromosome, and this locus here means the same location, so they can combine and work together and swap, and you'd code for the same characteristic. So you can get different factors that affect the production of an organism as well as genetically. So, for example, there's with a leaf, that's my picture of a leaf, you can have leaves that go yellow. This disease or environmental factor is called chloris. So when a leaf goes yellow, it has chloris, and this is due to the lack of chlorophyll, because obviously chlorophyll A is pigmented green, so that's what makes the leaf goes green. So when it has the lack of chlorophyll, it's an environmental factor that can affect the characteristics. So while genetics do decipher how many chloroplasts that can be produced at a maximum, the environmental factors including lack of light, mineral deficiency and infections also contributes. So while genetics deciphers how many of these chloroplasts can actually be made, the actual lack of light, so how much sunlight the leaf's getting, any infections and any minerals in the soil actually decipher how this is expressed, etc. So individuals of the species do have the same genes, but not always the same alleles. So they code for the same thing, so the same alleles, same genes, but not the same alleles in a species, because the alleles, you need variation. The genotype can define a child's growth. This is a picture of growth. So the genotype, obviously genetics, can define a child's growth of the maximum height, but the final height can be affected by diet. So that's another example of environmental factors. So while this child can have a maximum height due to genetics, it's to reach that large number, they have to have things like calcium, etc. And also I don't know if they have damage to the body. That's all environmental factors that inhibit the maximum the genetics provided. However, these environmental factors are not inheritable, so you can't inherit them. So such as a tattoo, etc. Environmental changes are called modifications, so it's been modified. Alleles here can be homozygous or heterozygous. So that simply means homozygous is the same allele, so 2 big A's, and heterozygous is 1A, little a. Homozygous and heterozygous. When two identical alleles for an organism's characteristic are identified, they're called homozygous. So as I've said there, two capital A's or 2 capital B's, so heterozygous and homozygous. They're also homozygous recessive and homozygous dominant. So say you have homozygous recessive, so that means you have 2 recessive alleles, so you can say little b, but they're both homozygous because they're the same, so that would be homozygous recessive. And then you can say homozygous dominant would also be the same, but homozygous dominant would be AA because that's 2 large dominant alleles. So heterozygous chromosomal pair is when there's 2 different alleles that code for the same characteristic in an organism. So heterozygous would be, as I've put here, big A, little a, or 2 different, 1 dominant, 1 recessive. But they still code for the same characteristic in an organism. So there are two types of variation. Just check. There's two types of variation. You can have continuous, or discontinuous. So the two types of variation are continuous and discontinuous. Discontinuous is always specific groups. So if you think of it this way, it doesn't continue, it's just discontinuous, so it's quite choppy. So examples of this is a blood type, so you can either have, I don't know, A, B, or O. I think that's what you can have. So you're always in one certain group. That's type of discontinuous. There's also there are also genetic make, for example. So if you have a wrinkled or smooth pea, that's discontinuous, because it can either be one thing or the other. So you can have a wrinkled, or you can have a smooth pea. That's a bad rhyme. A smooth pea. And then continuous, on the other hand, are values such as like height. So they can be like, it's quite a broad, it can be anything. Height, mass, so weight, and there can be a broader range of numbers. These tend to be a mixture of both genetic and environmental factors, as this is another example with the height, because it can have a maximum height or a maximum number of chloroplasts, but that to reach that maximum you have to have the environmental factors all also at a maximum to reach that highest level. So with the calcium, for an example. Additionally, going back to the leaf surface area is a continuous variation, as it can be any number influenced by both environmental as well as genetics. Sex, on the other hand, is discontinuous, so you could be either male or female. Well, okay, that can be debated, but you can either be born male or be born female. So that's discontinuous. So going on to a genetic cross diagram. When you draw a genetic cross diagram, there's a few things to remember. Number one, this is when you have a genetic cross diagram, is when you have you have the alleles, and say that's your alleles. The things to remember include, you always need to do the phenotype, which is the express characteristic of both of the parents. So say big A codes for blue eyes, so you'd put here blue eyes, and then you put blue eyes. You always need to put the phenotype and the genotype of both of the parents, so the parents. Okay, so once you've done the genotype and the phenotype of both the parents, then you go on to do the gametes of each parent. So these are the different combinations that you could have. So you could have here, you could have big A, little a, and then you can only have big A, little a, and two big A's, because that's what you can get here. And gametes, when you do the gametes, is always circled. So you have the genotype and phenotype, and then you have the gametes, which is circled, which is the combinations that you could have. And then you obviously do the Punnett square, which is you align it. So you do big A, little a, big A, and do the little square like that. And then you cross them up. Once you've done the Punnett square, you then go on to say the proportion of which of the genotypes are produced. So you say, so you could say here that, because this would be A, big A, big A, big A, little a. So all of these would be dominant, because they all have a big A. So it'd be dominant, so that would mean they all have blue eyes, because blue eyes was dominant. You'd also normally have if it was like half and half was blue-eyed, half was brown eyes, you'd have whatever the proportion was of offspring, and then you list the phenotypes that these offspring would have, and then you state the phenotype and the genotype of the percentage of offspring. So you would say would have A, which would mean it has blue. So you'd have to actually explain using both the genotype and the phenotype for both parents and the offspring. So a homozygous genetic cross, so say a homozygous genetic cross is something called true breeding. It's called true breeding. And this is for homozygous genetic crosses. So when you have big A, and then you could have little a, but these and these both need to be homozygous on their own. If two of these individuals with the same phenotype are crossed through several generations, the offspring will always have the same